Hi, it's Mr. Anderson, and in this video I'm going to go through the phases of meiosis. Meiosis is a lot like mitosis. It starts with interphase, but remember, the point of mitosis is to make two identical cells. And in meiosis, what we're trying to do are make four genetically different cells because they're destined to be gametes. They're destined to be sperm and egg. And that's the whole point of sex. We want the next generation to be different than the generation before. And so when you look at a diagram of meiosis, it's a little bit daunting. And we're not going to go through all of it right now. We'll go through this diagram at the end, kind of as a way to review it. But I want to step through each of those phases of meiosis. A quick mnemonic, PMAT times 2 is going to remind you the different phases that we have to go through. So it's prophase, metaphase, anaphase, telophase, and then we go through that again on the second division. Before we get to it, we should talk about the major structures that you're going to see as we go through meiosis. The first one, of course, are the chromosomes. And so you're going to see 2 of each chromosome at the beginning. And so we get a chromosome 1 from our dad and a chromosome 1 from our mom. We call these homologous chromosomes. Remember, each of these chromosomes has hundreds of genes on it. And so you get 2 copies of all those genes, one from dad, one from mom. This would be chromosome number 2 because it's shorter. And in my model, I'm just going to use 2 pair of chromosomes. Remember, in a real human cell, we're going to have 23 pair of chromosomes. But it's almost too difficult to follow what's going on if we had that many chromosomes. You just have to multiply it times 13. Now, lots of times you'll see chromosomes not look like that, but like this. And so what's happened here is that we've duplicated that chromosome from dad. And so these 2, what are called sister chromatids, are exact copies of the DNA. And so you know when you see a chromosome that looks like this, they've already gone through that duplication. Another important structure is the centrosome. The centrosome is going to organize the spindle, which is essentially dividing the nucleus and also dividing the cell. And in animals, it's made up of 2 things. We have the centrioles on the middle, and then these microtubules that go around the outside. In plant cells, they're going to lack these centrioles in the middle, and the nuclear membrane is actually organizing a lot of this division. But let's begin at the beginning of interphase. So this is just as the cell has been formed, and you can see right here in the nuclei that we have those 2 pair of chromosomes. Chromosome 1 and chromosome 2. And if we look at the centrosomes, we just have 1 centrosome. And so that's not usually what a cell looks like. This is what a cell usually looks like. And so what's gone on here, you can see that in interphase, we've duplicated those centrosomes. So we have 2 of those. And the DNA is all loose. It's not tightly wound up into these chromosomes that we characteristically see. As we go through interphase, what eventually happens is then we can see those chromosomes again. Now, do you remember what it means when they look like this X? It means that during the S phase of interphase, we've copied the DNA. So we have 2 complete sets of DNA. And this looks identical to mitosis, but it's just about to change. And so what happens next is prophase 1. During prophase 1, the chromosomes undergo what's called synapsis. And so what's happening is chromosome 1 from dad and 1 from mom are coming together. And they're wrapping around each other really, really tightly. And what's really going on is that they're swapping parts of their chromosomes. In other words, these are pretty much identical except for the changes in the genes. And so they undergo what's called crossing over. So segments of chromosome from mom are switching with chromosome from dad. Same thing over here, and same thing with chromosome 2. Now, what's that giving us? It's giving us variation. If this didn't occur, if synapsis didn't occur, the chromosomes that you get from your mom and your dad, you would give to your children, either as a chromosome from your mom or a chromosome from your dad. And what we're doing in crossing over is we're combining the 2 chromosomes that we got from our 2 parents and making a brand new chromosome that we want to deliver to our child. That's the importance of this. If we keep watching what happens next, we then go into metaphase 1. What's happening in metaphase, you can see that they're all lining up or meeting in the middle of the cell at what's called the metaphase plate. Now, they could have organized themselves like this, with the blue one on the left and the red one on the right, but they could have easily organized themselves like this. So they could have been in a different position. So this would be a totally different orientation of the chromosomes. It also could organize like this for chromosome 2, or it could organize like this. And so what do we have? We have 4 different ways that these chromosomes could orient themselves independently at metaphase 1. What is that giving us? Well, it's giving us variation. And so there are 2 raised to the number of pairs, possibilities of how they could arrange at independent orientation of metaphase 1. So how many are there? We would say 4. What if I had 3 here? Then there would be 8 ways that they could arrange themselves. Still doesn't seem like much variation. But remember, in humans, we have 23 chromosomes. And so there are over 8 million ways that all those chromosomes could independently orient themselves during metaphase 1. And so that's where we're getting, again, that and crossing over is giving us a huge amount of variation in meiosis. And remember that one sperm has to find that one egg. And so it's really over 64 trillion possibilities of an offspring just based on independent orientation itself. Let's keep watching then. So another thing that happens in metaphase 1 is that spindle is going to attach. So you can see that the centrosomes move to either side of the cell, and the spindle attaches to the centromere of each of those homologous chromosomes. During anaphase, it's pulling them apart. So we can see that those homologous chromosomes now are going to either cell. And then during telophase, what's happening is we're reforming a new nuclei at each side. We've divided the nuclei, which is meiosis, but now we have to divide the rest of the cell, and that's called cytokinesis. And so we're done with meiosis 1. We've gone through prophase, where we saw that synapsis. We went through metaphase, where we had that independent orientation. And now we've divided the nuclei into each of those cells. But we're not done yet. So what's going to happen next is we're going to go through prophase 2. Now, during prophase 2, there's no more crossing over. But what's going to happen is those two chromosomes are going to line up. Again, they're going to meet right in the middle. And the spindle is going to attach to each of those centromeres. Now, if we look what happens, each of those chromosomes are being pulled to another side during anaphase 2. And then finally, during telophase and cytokinesis, telophase 2, we've created these 4 cells that we wanted in the beginning. And so if we look at where did we begin, again, way back in the beginning, we had 4 chromosomes, or 2 pair of chromosomes. Now we have 4 cells, and each of those only have 2 chromosomes, a 1 and a 2. And so what would happen next in the circle of life? These would fertilize a sperm, or excuse me, if these were sperm, they would fertilize an egg, and then we'd start over again. We'd have a brand new organism that's going to be created through mitosis. Now, this is how we make the sperm. Each of the sperm are going to be made like this. Again, there'd be way more chromosomes in us because we have 23. It's a little different with the eggs because there's so much important material in the cell that only one of these nuclei will actually be used and the other ones won't be used genetically in that cell. And that allows us to keep all the important parts of the cell, like the mitochondria, endoplasmic reticulum, in that one cell. And so now let's kind of review and go over that confusing diagram at the beginning. So what are we looking at here? This would be interphase at the beginning. You can see we just have one copy of all those chromosomes. This would be at the end, so you can see that we've duplicated the DNA. Next, what do we have? This is prophase 1. To remember what important thing is occurring there, we've got crossing over, and then we've got independent orientation during metaphase 1. They then are pulled apart, and then we have 2 cells. And now in this second meiosis, what are we doing is we're just lining up those chromosomes, and then they're splitting up into each of the sides. And so what do we get at the end? Each of those cells, if we were to go back to here, each of those cells, look at this one, and that one, and that one, and that one, are totally different than that original cell. They also have half of the DNA that original cell did, and so that's meiosis, and I hope that was helpful.